In this video, I'm going to show you how to contribute to the Biomappings database of predicted and curated equivalences and other semantic mappings. The first thing we're going to have to do is clone the Biomappings repository off of GitHub. So I'm going to go into my development folder and I'm going to copy paste the git clone command. I've actually already got a copy of Biomappings loaded, so we'll move on from that. Next, I'm going to have to change directories into the Biomappings directory, and I'm going to have to pip install it. So I'm going to install an editable mode with -e, and we're going to install it from the source. And we're going to use also the web extras to make sure that we can install it with all of the things that are necessary to run the web curation app locally. All right, that's already done. So then I'm going to type in Biomappings web, and it's going to turn on the Flask app. So once we've done that, we're ready to go into our web browser again and load up localhost 5000. Once we've opened up the web interface, we can see that all of the predicted mappings are listed here. They've got source information, relation information, and target information with identifiers and labels. There's also confidence information attached to each prediction. This is a little bit different depending on which prediction method was used for each mapping, and that information isn't showed on this page to reduce clutter. But most of the predictions are from lexical mappings, and depending on what kind of prediction was made, the confidence may come from the lexical matching itself, or it may be an overall confidence assessment for the prediction campaign. That all being said, we're able to make some kind of filters or suggestions based on that, and then in the end we're able to either accept with a thumbs up or reject with a thumbs down. Note that most relations are curated, they're predicted as exact matchings. So we had a question about a specific thing called a bacteriophyllin. So we can use this Omni search bar just to look for any relationships that have been predicted for that key entry. So we can see that we've got this key entry. So bacteriophyllin is a small molecule, and it's been predicted with an exact match to prostatic hyperplasia. So this is a prostate neoplasm. These are obviously not the same thing, so we're going to be able to go and press the thumbs down button. If we wanted a little bit of extra information, we can click on the queries that have been hyperlinked to using the bioregistry to a nice source where we can get information about them. So for example, in this one, we can potentially read the description. We can look at some synonyms. So this isn't super helpful. Maybe BPH is why this was matched, because the other ones are very specific. On mesh, we can also look at some of the things here. So because B, P, and H are the first letters of one of the entries, it matches like it's an acronym. So this is not correct. We can also directly look at all of the different synonyms by expanding. In the end, we can come and click the thumbs down button, and it will reload the page, now with the curation done. In the background, the Biomappings web application is able to interact with the Git repository from which you cloned the Biomappings repository. And so it's actually made a commit already. So inside the web curation platform, we can actually have it interact with Git and make commits and pushes on our behalf. So we can look into my Git UI, and we can see that it took one of the rows corresponding to the curation we made from the predictions file, and then it put it into the incorrect file. So that's now sitting changed, but it hasn't been committed. So if we're inside our web interface, we can commit and push. I'm actually not going to do that right now since I forgot. I'm working on the master branch right now. Before doing all this, it's probably good to do making a new branch. So I'm going to start by creating a new branch, bringing those changes. All of this happens sort of seamlessly in the background, and now if I'm in the web application, I can simply press commit and push. It's going to take a second to interact with Git, and if we jump back into the GitHub desktop, you can see that there's been a commit made. So I'm actually going to have to push manually, but that's it. Now we're ready to make a pull request back to Biomappings to include our changes. Now we could potentially make many different curations all at once before making our commits and pushes. So we can also look at this bacteriophyllin A and bacteriophyllin. There's other sort of things that we can do. We can scroll through different pages. There's some other kinds of query parameters that we can add in as well. So I'll leave it at that for now, and happy curating.